नंदिनी, आप सुन पा रहे हैं हमें?
हाँ जी सर।
इंट्रोडक्टरी रिमार्क प्रोसीड करिए।
Am I audible and visible?
हाँ, हाँ, हाँ।
Hello everyone. I welcome you all for the 30 days international training come winter school. A very warm and lovely good evening. I am Nandini Sharma, your moderator for today's session. And the topic for our winter training is navigating agriculture transformations for the upliftment of rural excellence. That is Nature 2024. Today is 16th day of our training, organized by AgriMeet Foundation Bharat in collaboration with CIMMYT Mexico, Southern Federal University of Russia, ICARDA Lebanon, World Agroforestry Center for International Forest Research Kenya, ICRISAT Hyderabad, Manage Hyderabad, SKL TSHU Telangana, MGUVV Durg, HAU Hisar, IGKV Raipur, IARI New Delhi, MHU Karnal, NDRI Karnal, ICAR New Delhi, ICAR DCR Puttur Karnataka, Stanford University, and University of California. It is my immense pleasure and privilege to extend a warm welcome to all of you on this special occasion. Today, we are honored to have with us a distinguished scientist and a visionary leader and a pioneer in agriculture research, Dr. Manish K. Pandey, sir. Dr. Manish sir is currently leading the groundnut and pigeon pea genomics pre-bridging and bioinformatics group at the prestigious International Crop Research Institute for the Semi-Arid Tropics, ICRISAT in Hyderabad. His exceptional work in developing and deploying genomic resources for breeding climate resilient and nutritionally rich crop varieties has had a profound impact on global agriculture. With more than 200 scientific publications in high impact journals such as Nature Genetics, PNAS USA, and Molecular Plant, and over 12,000 citations to his credit, Dr. Manish Sir's contribution are a testament to his unwavering dedication and expertise in his field. His achievements have been duly recognized as he has been inducted as a fellow of several esteemed scientific academies including the National Academy of Agriculture Sciences, New Delhi, and the Telangana Academy of Agriculture Sciences. Dr. Manish Sir's groundbreaking work serves as an inspiration to all of us, especially in times when addressing the challenges of climate changes, food security, and nutrition has become more critical than ever. We are truly fortunate to have him here with us today to share his invaluable insights and expertise. I am confident that his guidance will inspire and equip us to contribute meaningfully to the advancement of agriculture research and practice. On behalf of everyone present here, I extend our heartfelt gratitude to Dr. Manish Sir for gracing this occasion with his presence. We look forward to an enlightening and enriching session. Once again, a very warm welcome to you, Dr. Manish Sir, and to all the participants. Let us make this day a memorable and impactful one. Over to you, sir. Session is yours.
Thank you, Nandini, and thanks to Dr. Ankun Sharma, for approaching and then facilitating all these presentations. Good evening, everyone. I'm happy to share some of the experiences that I had in last 1 decade, and maybe I would like to share like where the genomics is heading towards maybe next 5 years or so. Let me share my slides. Please let me know if once you see slide.
Yes, sir. Yes, sir. It's visible.
Okay. Okay.
यदि सर पॉसिबल हो तो इसको पीपीटी मोड पे कर लीजिएगा प्लीज।
Okay. Let me do that. 1 minute. Issues. Can you see slides?
स्लाइड्स विजिबल है सर, लेकिन पीपीटी मोड पे नहीं
Oh,
है। अगर आप ऐसे कंफर्टेबल हैं तो प्लीज आप प्रोसीड कर सकते हैं सर।
let me try
हाँ
once
यस।
more. So
डन सर,
still not in. It is
डन
okay
सर,
now?
प्लीज प्रोसीड। हाँ डन सर।
Okay. Thank you. So, what I would like to talk today about the topic, I think you are all aware about the gene bank genomics and pangenomics for crop improvement. So, let
आई थिंक कोई
uh,
टेक्निकल ग्लिच आ गई सर के एंड पे।
so PPT is not coming now.
हाँ नहीं सर, पीपीटी नहीं आ रही है।
No, sir.
एक बार रीशेयर करिए सर प्लीज आप।
एक minute. Just give me a minute.
हम्म।
Now, whether you can see slide?
यस सर,
Yes,
विजिबल।
sir.
Okay. So I think many of you know about the ICRISAT, which is based in headquarters is based in Hyderabad, but it has a country office West Central Africa. And here we have a genomics lab that you see Center of Excellence in Genomics and Systems Biology. We have a small group also here that work in this crop. Sorry, something.
सर आई थिंक नेटवर्क का इशू है, क्या है मैं समझ नहीं पा रहा हूँ।
Yes, sir. Network issue लग रहा है. Video off है.
नेटवर्क इशू लग
Yes, sir.
कोई ना, रिजॉल्व हो जाएगा सर, कर लेंगे उसको। रोहित आप देखिएगा थोड़ा सा, ठीक है।
Yes, sir.
हम्म।
It seems some problem in
सर यदि पीपीटी शेयर में कोई ग्लिच है तो काइंडली आप पीपीटी मुझे भेज दीजिए, मैं अपने एडमिन से शेयर करा देता हूँ।
Okay, let me try once this time, and then if not, then I will share. Hope this
जी
time it works.
जी जी।
Sir, while sharing the PPT, I think, yeah.
Coming?
Yeah, sir. Coming, sir. Sir, not in PPT mode.
Now you can see the slide.
Slides are there, sir, but not in PPT mode, sir. Like normal are there. Yeah, now it in PPT mode. Can you just move 1, 2 slides, sir, so we can show that it's working?
We do that. Yeah.
No, sir. Not.
No, sir, I think it's not working in PPT mode, sir.
Should I share you now?
हाँ सर, हाँ
Yes, sir. Please.
Give me a minute.
It's moving. So I think, sir, the network issue. I think it's okay, sir. Just share the PPT with us. We can do it from our
Okay.
end.
I think to remove such type of glitch, PPT share will be the good option.
Yeah, yeah, yeah. Always that's good.
हाँ डॉक्टर रोहित, आप अपनी तरफ से इसको प्रेजेंट करिएगा, ठीक है?
Just
आप सर मुझे, मैं आपको ईमेल दूं कि सर व्हाट्सएप कर देंगे आप? रोहित, मैं व्हाट्सएप करूंगा आपको, ठीक है?
okay, sir.
So this is bigger one. So what I will do, I will save in PDF mode.
ठीक है सर, आप कन्वर्ट करके भेज दीजिएगा।
No
Yes,
issue.
yes, yes.
सर आप मुझे व्हाट्सएप करेंगे कि मैं ईमेल दे दूं अपनी आपको व्हाट्सएप?
उसमें भी कर सकता हूँ मैं, मे बी।
हाँ ठीक है सर, व्हाट्सएप पे कर दीजिए, ठीक है।
Please check once.
जी सर, आ गया। रोहित जी, मैंने आपको भेजा है, आप
Yes, sir. It's moving, sir.
Yeah. Thank you.
Yes, sir.
So maybe we can go to next. I was just talking about that many of you know about the ICRISAT, and I was telling about the gene bank. Yeah, please be maybe previous slide. Yeah. So we all are aware, like if you see globally, there are like around 11 CGIAR gene banks. When I say CGIAR gene banks, those are for different crops. For example, ICRISAT is one of those CGIAR institute, and it has around 120,000 germplasm in its gene bank. Now, what I mean to say here that for most of the crops we grow and we eat, we see that the diversity is existing or conserved, you can say preserved very well in these gene banks. And not only these CGIAR gene banks, the diversity is conserved in many of the national gene banks, including India, that is one of the like say largest gene bank in India, NBPGR, that holds millions of samples. Next slide. Now, what we know that during the domestication, I mean to say the agriculture, what you can say breeding that has started maybe as a half a century or you can say few decades. But what was happening actually from the 5,000 or 6,000 years back, what was happening till that time? Actually, it was all about only like those farmers, what you can say farmers or the like say our predecessors, basically those ancestors, you can say they were conserving those germplasm and they were actually indirectly selecting some of the traits. It can be seed size, it can be germination percentage, it can be flowering duration, it can be plant height. So I mean to say the major traits that have been knowingly unknowingly been selected that time also, but in a long duration. But what has happened that if you see here the crop wild relatives that has large number of diversity, you can see different colors, their symbols. But when this cultivated, I mean to say, if we take example of rice, wheat, maize, or anything that has been cultivated, then we lost diversity. And where the diversity is, that is still in the gene banks. But whatever we eat, actually, we grow in the farmer's field, that is like a fraction of diversity that we are growing and using as of now in the farmer's field. That means when we are seeing the climate change coming up, the already like gene banks store all those genes that can support our like say breeding future ready crops. When I say future ready crops, that means they can sustain to the climate change problems in future, whether it's a heat tolerance, drought, or any kind of other stress. Next slide. Now, what we have been doing, how we have been using genomics, if you see in this slide. So you see genetic resources, I mean to say global germplasm collection that I was talking about just like I said 2 slides back. Then from those germplasm, we have mini germplasm sets, I mean to diversity panels, which we have been using for phenotyping and then sequencing and all those things. Then genetic populations, not only germplasm, because the concept from the natural population association mapping came from the human science, because their family-based population development is not possible. But in plants, we have that flexibility. So we have been many of the crop plants, you might have heard about the family-based genetic populations, whether it's a recombinant in bed lines or it's like nowadays multiple parent, multiple parent advanced backcross lines or anything you can say magic and all those things. Now, what we need actually, we need a sequencing, genotyping, and also we need precise phenotyping, and then we can associate. But for that sequencing and everything analysis, gene discovery, you need some of the basic genomic resources. I mean to say, if you want to do for SNP calling, you want to know where the genes are located, you need a reference genome. But nowadays, not only just one reference genome, we see that core and pangenome. That will talk these things in later slides because that is the presentation on. But what we have been doing, we have been developing different, like say using these resources for gene discovery, assays, all those kind of applications. Now, what we are using in the breeding actually, what we are doing actually using these genomic tools, then we can identify duplications in gene bank, we can study genetic diversity, whether it's a complete gene bank collection or
रोहित जी क्या हुआ, आवाज हमें ही नहीं आ रही कि आपको भी नहीं आ रही?
नहीं आ रही सर, शायद डिस्कनेक्ट हो गए सर। कनेक्टेड तो है सर, हाँ डिस्कनेक्ट हो गए सर।
आप देखे रहिएगा, तुरंत को होस्ट बना दीजिए।
Recording in progress.
I think there was some misconnection, but now I think it's so voice may be coming. Can someone confirm once?
Yes sir, आ रही है।
Yeah.
Yes.
So then I was talking about this linkage drag. That means when you are crossing with the y-axis, you can increase, I mean to say reduce the linkage drag. Hybridity confirmation in hybrid crops, many of the crops you find very difficulty like to see whether the F1 is correct or not. Then you can check the hybridity and you can confirm very easily. And nowadays new application is coming for the seed system because you see like there are mixtures there, so you can identify with marker tools. Next slide. Now, I was talking about reference genome. So just I will give example in groundnut. So many of the examples I may give for groundnut because I have been working since a decade. So we developed like a reference genome high quality for the diploid porogenitors and also the wild tetraploid species. And that is like common in every crop. So there is nothing new about it. So you may be finding multiple genomes now in different crop species. Next slide. Now, in this slide, just I would like to mention one point that the genomic technologies have helped us really in developing even the complex genomes. Why I was talking complex? Because groundnut itself is a tetraploid where two different genomes are living together. And they're really like 90 to 95% similar with each other. So it was very difficult actually to differentiate. You can say just a few percentages of difference between these genomes. So you need to use like say the advanced technologies, I mean to say Hi-C, PacBio and BioNano also, so that you can have basically a very high quality reference genome. So that I wanted to mention here. Next slide. Now what we do with these genomes, ultimately for the breeding, for the crop breeding, you need genotyping assays. I would like to say here for a few examples using those genomes and resequencing data of 300 genotypes. We developed high density chip that is 58,000 SNP. You can see in the left side that is there in Thermo Fisher platform that you can use for fingerprinting, genetic diversity and trait mapping and also for genomic selection. But the cost of this chip comes around $55 to $60 per sample. Now we have mid-density panel, next one that we developed with Rosenberg Institute, and that there are 5,000 SNPs from the genome wide, and that is also on Thermo Fisher platform. But the cost here is just $8 per sample. So you can get 5,000 SNP data in just $8. Now the next panel what we develop is low density, that is 20 SNPs that you can use for hybridity confirmation or just some genetic impurity, you can use those markers. So I mean to say these different density, they are of different applications. So we need to keep in mind the cost, because when you need large number of samples, we need to reduce the number of SNP so that it can become cost effective. That's the message here. So if you see the 58,000, we are investing around, say, maybe you can say same $58 per sample, but when it is coming to 5,000 SNPs, we are spending $8 per sample. But when we are using just 20 SNPs where it is needed, maybe for foreground selection or so, just $3 per sample. So that is how it works like different platforms. And we are still trying to make these genotyping assays as much as cost effective so that it can be adopted in large breeding, like say programs. Next slide. Technologies I was talking about in the previous slide, we are putting all the information in the EIB website that you can see here, not only for groundnut, you can see for multiple crops. So I think you may be interested in maybe some of these crops. So if you need any kind of information, whether mid-density or foreground markers, something, you can search this website. These are available and you can get genotyping data. These are in open genotyping platform. So anybody can access even from remote areas. And the mid density that we have 2000 and the 20 SNPs, even you don't need to isolate DNA. Actually, you just chip leaf samples from the field and send samples, you get genotyping data. So that kind of approach, because we were working like say finding difficulty in African locations in the remote areas where DNA isolation facility was not there. So we have to come up with such technology where DNA isolation is not required. Just we can send the leaf samples and we get the genotyping data. Next slide, please. Now I'm giving just example for groundnut. So once we do trait mapping, you see here product scoping. I mean to say the traits mentioned here, rust resistance, stem rot, and all those traits you are seeing. What we do, and in the breeding program, we prioritize traits. We don't try to develop markers for all the traits. We discuss in the the traits which can be easily phenotyped, we don't worry about, but where the time and money, the resources are more used in phenotyping and finding difficulty to phenotyping, then we are working on trait discovery. So you can see here already we have developed, if you see S4 trait deployment, already we developed markers for rust, LLS resistance, high oleic and fresh seed dormancy, and the markers for blanchability and aflatoxin, those are in validation. I meant to say you can just use around 20 SNPs and you can select 5 to 6 traits in one go. That kind of application here. Not only these are used, you can see right side the countries where these SNPs are getting used. And it's not like we, these information, whenever it is there, we are validating, we are making available in the website that I mentioned just now in the AIB website. So anybody can access that. And there are few examples. Maybe if you go to next slide, can you go to next slide? So these markers were used in India, and you can see around 5 to 6 varieties are already released using these markers. So you can see the genomic, and you know, if you want to see what kind of benefit you are getting using these markers, so it can reduce the, like say, duration around 40% in developing the new variety. If you want to just integrate, I mean to improve the traits that are listed there and markers are available. Maybe not now, but if you see, take example of rice, there I think you have for many traits, those such markers are available. So you can select many traits just using by genotyping. You need not to phenotype. Even for groundnut, actually, suppose if we are genotyping, let's say F2 population, we need not to plant, we just chip the seed and just send for genotyping, and once we know that the desired alleles are there, then only we'll go for germination and planting in the field. So that's how we are operating at Acrisat, and we are operating in a way like around 10,000 samples per year kind of thing in groundnut right now, for and like this operation I was mentioning. Next slide, please. Now, how these have become possible? You all are aware about the sequencing platform. So it's like you can see, you can compare to the like say transport. Earlier we were traveling on like say trains. Now we have speed trains. What does it mean? Actually, it is saving time, and that is the main thing that we want to save times, because the climate change is happening very fast. So our breeding program needs to match that speed. Otherwise, we will be left behind. How these sequencing technologies are helping us? If you see, like in the over the time of maybe 10 years or 20 years, the throughput has increased. I mean to say these machines can generate much better, higher data in less time. And the second application is this seed, I mean to say the sequences that is coming read length. That means even like even say you want 200 base pair, but now you can get up to 10 kb when you see in the different platform, the third generation sequencing that we are talking about. And you can get from PacBio or Nanopore, you can get that kind of like say bigger sequences. Next slide, please. Now these technologies, next slide, please. Yeah. So when you see the cost and the times, you see the very good graph here that is coming from the human sciences. And that is how the change that they have brought from, you can say in last 20 years, huge reduction in the cost, because once you have like more applications, the throughput and all those things, there is a competition between companies, and then as a customer we are beneficiary for these things. So really that is been drastically reduced. It's very much affordable as of now, so which can be deployed in every research teams irrespective of size and the budget constraint we have. Next slide, please. Now, what is happening once you see here, if you see again what I was saying, that length and technology and the NGS technology, Sanger, that was long back, but you see the 10x genomics, BioNano, and all those things, PacBio. You see the throughput, I mean to say the sequence length had increased. That has helped actually in developing reference genomes, even for the most complex genomes like wheat. That is really one of the most complex, and also in size, the huge sizes. Next slide, please. And what you do with those data sets. So these sequencing technologies have helped us in understanding many things that was hidden information in terms of genome. Number one is evolution. For each, now you can see, you know that those are the from the previous evolution. If you see, so initially we are only 7 chromosomes. Those were the, like say, you can say founder genomes or any genomes, you can say those ones. And from those 7, all the crops have evolved then, and the chromosome number and then the sizes have become different because of duplications, insertion, deletion, and all those things. And that is what made the each crop is special, each species is special. But how we understand this? So sequencing technologies has helped us in identify. If you see right side, those colors different, you can see from the ancestor genomes how which chromosome is similar, which part is similar. So you can see how the genomes of a particular crop is evolved. Here the example is groundnut again here, and you can see how much similarity they have from different related genomes or even like say distant genomes. So you can see, so that is like say the basically benefit of sequencing technology. Next slide, please. Even now, we know that the crop is very domesticated. If you genotype or sequence the germplasm across the world, then you can also come to know that where this crop is basically originated. And then what is the proof? Proof where it has moved from one place to another, what kind of changes accumulated during domestication, and then travel of these germplasm from one continent to other continent. If you see the right side picture that is mentioned here, it is clearly stated that the origin of peanut that is there in somewhere in Peru and Bolivia, all those border near Argentina. So you can see where it is originated. So you can trace down origin. That's one of the basically beauty of this, and that is one of the example of those. Next slide, please. Of course, this is, I will not take here time. Everybody is available. You can do diversity even with 50 markers, even with 10,000, even with million number of SNPs. But that is how as much number of SNPs, as much reliable the genetic diversity results are. Here, see from the sequencing data, you can clearly see, you can differentiate the elite, land races, breeding, or even if you have some accesses unknown, you can figure it out whether it belongs to elite line, land races, or breeding line. So like in the gene bank, if you get entry and somewhere you are lost, that there is no name or some identity is lost, you can sequence them, genotype them, and then when you compare with the rest of the germplasm in your bank, you can clearly put them in the order that, okay, this may belong to this group. So you can have some idea on those things. In right side, you can see clearly different groups: Fastigiata, Hypogaea, vulgaris, and Hirsuta, all those groups separately. So that means these groups, they conserved specific genomic regions, which are helping actually when we are sequencing to make them different groups. So you can understand if you want to bring diversity from one group to another, you can do it. Next slide, please. Now, again, this is one tool from the NVPGR India. You can see if you have if using this tool, you can genotype the germplasm, and also it can detect duplications. That is one of the beautiful application of this. This is one, and the paper is given if someone is interested to read it. And you can just there is like algorithm already, and it will say the duplicate if like the difference threshold is of 0.1%. If that much similarity is there, it will tell whether it's a duplicate or it's a different genotype. If it is a different genotype, how much diverse it is from rest of the genotype. Next slide, please. Here I'm just talking about that is one of the gene bank which is being sequenced, that is for barley by IPK Gatorsleben. And you, of course, this is one of the examples. You can study population structure, and within population groups, I mean to say, you can see the admixtures. I mean to say, within a group, all the lines in a group may not be true to that group. So it will have different levels of admixtures. Few lines will be there 100% for that group, but others will have part of their genomic regions matching with other different groups. That means the diversity you can see within a group, how the group is diverse, and the particular line is having blood similar to other groups, so that you can compare. So that's the goodness of this sequencing technology. Next slide, please. Now, the question here is, now if we don't use genomics, let's put it together like that. We are like maybe there is one scientist 20 years back, and the person went and collected accessions from different parts of country and put in gene bank. Now again, after 20 years, somebody is going again, next scientist, and again bringing germplasm from maybe the same location, we don't know, or maybe nearby location, but we don't know whether the collection is that already we have in the gene bank. It's very difficult to say. So what happens if you genotype, then you come to know whether the like among all corrections, I mean, say, let's say example, in ICRISAT, we are trying to genotype all the 15,000 germplasm. We may come to know that it's not 15,000, maybe it's just 12,000 accessions are unique. The 3,000 are just duplicates. They are collected by different time people and maybe shared by different groups. And those are conserved in the ICRISAT gene bank. And what we have seen from the previous experiment, that is from coming from WGRC, CIMMYT and PU, then 26 to 54% redundant accessions are there in collection. So we have to see each collection, there may be a potential that such things are happening. And so each gene bank, if you want to reduce the number of germplasm, I think this is good way to genotype them and just identify duplicates and then reduce the collection. So it will reduce, I mean to say, the investment that is required for those extra redundant accessions in the gene bank. Next slide, please. Now coming to the pangenome, what we are talking actually. Let's now, before going details, I will just want to give example. If in India, if you develop a reference genome for one person, let's say Dr. Ankur Sharma, and then you sequence different, like say participants, let's say 200 here, and we map our genome to Ankur Sharma's genome, I doubt that all our genome, I mean to say sequencing data will be mapped to him, because each of us carry some of the unique genes. And that's what make actually the different part. So when all our genomes, when they are like say matching, that is like a part of core genome, and then others are like unique genome. So that kind of, and like say you can identify how much similarity and all those things. That is one of the application. Next slide, please. Now, pangenome is not a new concept. Basically, it has been getting done, and it started from 2005, but we never thought of that. The technology was not cheaper at that time. So mostly we saw either for the microbiomes, because the microbes have a smaller genome size. But now what you see when the as per the, like we see the genomics technologies, I mean to say sequencing technologies evolved, we have been seeing for the pangenomes for the complex crops and crops of the bigger genome size. I will give many of the examples in following slides. Please go to next one. Now, how you are going to develop. So like you may get to, maybe you can read about this paper. This is one of the good paper to read about what is the pangenome and how it is developed. In a nutshell, I can tell you, to and also clarify that when I say pangenome reference means that you develop many individual de novo genome assembly and then club them together in a single reference genome, then you call pangenome reference assembly. When you have only one reference genome and you do just 10x coverage of, let's say, 200 accessions, then it will be like pangenome kind of development here, mostly that we follow terminology as of now and in this particular arrangement. So if you have only one genome, let's say from Ankur Sharma, then we are aligning all the 200 our genomes, so we can develop pangenome. That means we can develop a pangenome, whether it's a iterative based or graph based, where we can add all our additional genome sequences, unique sequences to the Ankur Sharma's genome, so that, and his genome size, I mean to say that outcome will be bigger genome. That means all our diversity will be captured. So whenever we are doing further analysis for SNP calling and all those things, no diversity will be left out and you will get SNPs for all those genotypes. Next slide, please. Now, once you develop this pangenome analysis, that means you are capturing the diversity more than one genome. It depends how many. Each study you see, it will report different number of genotypes. So, and it is very much like, say, pangenome is very much like, say, related to number of genotypes, accessions, assembly you use. That please remember that. If you are having pangenome from 200, it will look like different. If you have pangenome analysis, or let's say using 200 genotypes, that will be different. If you use all the 15,000, that will be look completely different. So that's how it is. So it depends on the number of genotypes you are using. Please refer to always it's relative as much you can. So earlier we are like a few pangenome analysis were conducted with maybe 100, 200, but now the size is going to the 3,000, 5,000 accessions as of now. I will give few examples. And what it is helping actually, it is able to capture the diversity, entire diversity, so that none of the diversity is left out while we are calling SNPs, we are performing GWAS analysis or annotation, anything. So that is how it is helping us. Next slide, please. And if you see, even like the different ploidy level, this is from the potato species that you see, that even like the crops where you have ploidy, I mentioned whether it's a allotetraploid or autopolyploid, you can do all those things. And here you can develop like identify core and pangenome. This is same thing what I was saying, just showing you few examples of that. And you can also see that separately diploid and polyploid, what is the status, all those things, where they are, how much similar they are. You can get all those information from this. Next slide, please. This is from the Pangea, you see, genomes you can see, and you can see how many samples, just 27 samples, and you can see how many percentage genes, all those you are adding, from how many are there genes when you have 99 samples. You can see all those scenario, I mean to say, and then you are adding actually new variation to your analysis. So that is, this is from the MAGE. Can you pangenome? Can you go to next? This is, I'm just like bringing up here from the pathogen here. This is A-flavus. This is one of the, like, say, fungus. And like one of the study in US that we have done together with them, like 346 from different states of US. And what is important was in this pangenome analysis, that there was one state where the toxin production of A-flavus was very high. So there was no information, where there was no region actually identified why this was happening. Later on, we identified that there are actually some of the genes, they have accumulated more mutations in a particular region where the drought and heat was high. So pathogen evolved, and though that evolution actually led to higher toxin production. So I mean, so that we come to know because of the pangenome analysis that we perform. So this is one of the, like, say, good example of that for the pathogen, how we can use to like, say, enhance our understanding on the pathogen evolution. Next slide, please. is pigeon pea pangenome. So initially it was developed, just using 89 genomes. If you go to next slide, please. Now we are working on improving this pangenome, and now we are using around 400 pigeon pea genomes in that one. We're using this iterative mapping assembly that we are working on. So now we are adding up, many of the new sequences to it, and the pangenome assembly
शायद फिर नेटवर्क चला गया है सर का। रोहित,
and then
Hmm.
I think there was some problem. It went off. Then you have chickpea pangenome, and then you can see here, like you can identify desi and intermediate Kabuli wild and all those things. Very just example. Can you go to next slide, please? Then you have here Brassica oleracea. You can see pangenome from, you can see different hotspot, and you can see here from the picture those a good analysis where you can identify this cabbage, even like the Chinese one, and broccoli and cauliflower. Really, this is very beautifully study. You can see how it is differentiated. We all are aware about this Brassica triangle, all those things, study those. And see, this is one of the pangenome that has helped really. You can see here early domestication, that is, you can was identified because of this pangenome analysis. You can see those colorful regions, those have gone under domestication, and you can see clearly those domesticated regions have changed the entire look of the this Brassica, I mean to say, family. You can say in terms of those are looking completely different, and we use them in our different preparations for food. Next slide, please. Now this is rice super pangenome, if you see, and 251 accessions, and this is also coming from the different parts of the world, and you can clearly identify different, like whether it's a Sativa indica or Sativa os or Japonica, Rupipogan, all those things you can differentiate. This is one of the good example. Next slide, please. Here you have for the grape, so it's a similar, you can see pangenome here, core genome, dispensable genome, private, I mean to say, those are the unique. Sometimes authors use different words, but the private means is whenever there is unique, those genome. So otherwise, pangenome, core genome is same, that is similar to everything. And you can see different species here. So there are some genome that is core, that means that is present in all these species, but there are something that is really, I mean to say, unique to those different species. Next slide, please. Now this is chickpea super pangenome, you can see, and here you can see different wild succession. If you see Aritinum and all those reticulatum and all these echinospasm. So basically you can see different species here, and this is published in 2024, and you can see that how much core, how much dispensable, and how much gene families are there, and also like structural variations, how those are different in different you can say species. And also see how these species are also evolved. So that's also one of the things that you, among wild species, you may come to know that which wild species is more say nearer to the cultivated ones, which one is like say more distant, I mean to say different from the cultivated ones. So all those information, and once you are trying to progress basically wild genome into cultivated, you need all these genome assemblies of wild to perform analysis. So that's also requirement. Next slide, please. Next slide, please. Here you have tomato super pangenome. So I will not go detail in terms of time, but you will find I have mentioned those references sometime. And these slides, Dr. Ankur Sharma, if you want, participants can be shared, not a problem with me. So here you see all those scenarios where from the different 11 distant tomato species this is developed. So it's just like 8 from the chickpea, 8 or 10, but here you can see tomato 11 such species. So this is similar thing, like you can identify all those things. This is just an example. Next slide, please. Now this is watermelon super pangenome. When I say super pangenome, that means it is like bringing different wild species here. So let me see clear here what is this pangenome and super pangenome. So suppose if you are just having one species, let's say I'm talking Arachis hypogaea groundnut, and if you are sequencing these accessions from the same species and you develop pangenome, that is just pangenome. But if you are sequencing different species accessions and you are doing performing analysis, I mean to say, like the example you have seen in rice, tomato, all this chickpea, 10 and 11 different species, then it becomes super pangenome. That's the difference in terminology. Not to just wanted to clarify, not to confuse here. And here the assembly were used really very good because these were telomere to telomere. Assembly, so that was really very good for this watermelon. Next slide, please. You have for the populus, we all know about the utility of the populus tree, and then you see the pangenome from different species, and then this is come in 2024 this year itself, and from it was from 19 populus genomes. What I am saying here, that in future we may not have just one genome in each crop species. We may have maybe even like say 50 to hundreds of individual reference genomes in crop species. And also for other, I suppose if you have interspecific, I mean to say different species, everything that will become available maybe in 10 years. I'm not sure if the technology is going in the same way. Each individual, I mean to say, of the accessions germplasm in gene bank will be sequenced in next 10 years. That is how the technology is going, and maybe we will have much more better analysis and understanding on these genomes. Next slide, please. Now, the clear message is now whether we are using moving from genomics to pangenomics to some extent, yes. We are using, there is a bias when you use single reference genome, whether it's a SNP calling, trait mapping, or any application. But when we have basically diverse reference genomes, then it becomes very good that to use all those reference genomes into a pangenome assembly, and you perform analysis. So really that is going to be like say moving ahead. That is the way doing in future. Yes, accessory genomic regions should be the important traits. That's very important from some individuals. That is what's completely missing. For example, in one of the experiment, when we were mapping to the reference genome of Arachis hypogaea, maybe 1 to 2% was not getting mapped. But the genes that was not getting mapped, I mean to say sequences, the resistance genes were lying in that region. So that kind of thing may not be happening when we use pangenomes in future. And of course, abundance of non-coding disease-associated variants are also getting identified. We are learning many things from the human genetics, and we are trying to apply in the plant. So I think that is one good way. And really, when you see pangenome and sequence and alternative alleles in the sequencing, so it's very important to see that accuracy. And also this, when I'm talking pangenome analysis, so initial phase of pangenome and genomics has been on short reads. But what I'm saying that in future, these long reads are mostly used now, but I think it will increase. For example, if you see in our genomics studies, right now you are using 80% short read data. Let me put like from Illumina, and also like 20% only from PacBio or something. It will reverse in next 5 years. 80% may become like say a long read and 20% from the short read. So that's how it may go in future. And we also see in our genomics study as well. Next slide, please. Here, just wanted to put together that when we are generating large data, whether it's a genomics data, here's what we discussed here, but also in the parallel, the data is generated on transcriptomics, proteomics, and metabolomics. Once you have all those data sets, I think we need artificial intelligence, machine learning algorithms to understand. Manually, it is just can't be managed. So what I see in future, there is like we need to start working on this aspect, learning this AI ML applications, maybe even small, but maybe in 5 years we will learn more. So I think I will, if we, the groups, there are youngsters, new generation of scientists, I will just suggest that please get into hands on these. This is the like AI ML is future when you will be analyzing the data, because we are generating huge data. It's a problem now becoming not data, but how to store it, how to analyze it. That is the bigger problem. So we need all these applications. Next slide, please. Now coming back to the same slide, that during domestication breeding, we lost many of the diversity. Now it's time to characterize well those germplasm traits and use the genomics, bring back those useful alleles from the wilds to the cultivated background. I mean to say the modern varieties that we developed in future, let's use the technology to make them more climate change proof. And so we need the useful alleles from the wild. We need the help in breeding future varieties. Next slide, please. Now here, like, just wanted to mention one of the not just PacBio and Nanopore, but optical genome mapping, that is also there, where you can even like you can get even 300 KB data. So when you are interrogation, especially, it is has been heavily used in developing reference genomes, one of the technology, but also it has applications in pre-breeding where you can trace down the larger gene duplications, inversion, deletion, and all those things in your pre-breeding populations. Next slide, please. And what I was mentioning, the long read, what they have helped, how they have helped from the small pieces, earlier short read, just you can see left side. And if you have like 10 million pieces and 2000 pieces, you know that how easy to solve the puzzle pieces if just we have 2000. So for schooling, it is easier if you have long read data. So that is how the this has improved. Next slide, please. Now, just not only for the next slide, please.
रोहित जी, आप सुन पा रहे हैं?
सर डिस्कनेक्ट हो गया सर।
कोई बात नहीं, देखे रहिएगा, जैसे ज्वाइन करें तुरंत को होस्ट बना दें। Yes, so you can see like how it is happening. You are seeing even transcriptome analysis. So it's not genome. I mean to say, when you have bigger pieces, it's very easy to go for like EG analysis with the long read sequencing data, and the accuracy is very high. So this is going to happening in all the I'm not saying right now it was happening only with the reference genome assembly development, but now it is happening in transcriptomics data or genomics data, and in future it will increase more. Next slide. Even like you can say structure variations here. This is example from Brassica napus, and you can say vernalization genes are identified. This is one of the very beautiful examples coming here. Please go through it. When you do all these analysis, you identify big chunk of amino inserts and deletions. So that is one of the good example. Next slide, please. Why multiple genomes are important? Already I told you before, I mean to say in previous slides, but here I want to say that each genome is carrying unique genes that is very much required for the analysis, and that is why multiple genomes are coming. Next slide, please. There are few examples I will be giving the multiple genome assembling in soybean. Maybe because of time and all those things, little bit will skip. You can go to next slide. Here the 26 soybean genotypes were used. But if you see the beautiful picture, if you go to right side, you see how those different genomes, different genomic regions, they are different. And you can really come to know how these individual genomes are in a way functionally different. You can get to know if you have trait phenotyping data. I mean to say if you have different high oil content, or you can say different plant type, you can easily get into all those domestication related traits very easily. Next slide, please. Then what is happening in humans? Actually, this is one of the examples. The paper came in 2023. You can say two groups, one from the US, and they have like basically selected samples from like you can say 47 from different countries and developed one pangenome assembly. When I'm saying pangenome assembly, again, whatever I said, the individual genome assembly of 47 genotypes, they were clubbed together. Here this is human, so I mean to say you can say from the 47 humans, but their goal is actually to go to 350 human genomes. Maybe I kept mid 2024, I have to see whether it has been completed, but definitely this is 350 genomes together. That will be very huge. If you see large side, you see that is paper coming from China. So they have like they had 116 high quality genome assemblies from different states, from different tribes, and based on the genetic diversity they have done, and they have developed pangenome reference. So this is in human science. I think this is the same thing going to happen in crops as well. I mean to say we may see in a particular crops that there may be 100, 200 individual genome assemblies are developed, and those are being used to assemble into one, that is called pangenome assembly. That will happen. Next slide, please. What we are trying, suppose here see groundnut, you can see pigeon pea, there are different species, subspecies. In the center where the primary gene pool is written, that is we grow actually in field, farmers field, but rest of the species are wild, and really those are untouched. So there is a lot of opportunity to sequence them, understand them, and develop pangenomes, and then utilize them in the breeding program. Next, please. What we are trying here actually, that suppose if you have 15,000, I'm just giving here example for pigeon pea, we are growing all the 15,000 germplasm. We through speed breeding, we are selfing for 3 generations, because this is a partially you can say cross pollinated, and then we want to genotype them all, group them into different groups and subgroups, and from each group and subgroup we will take one representative genotype to develop individual reference genome assembly. And suppose let's say we get total 27 such groups, subgroups, that means 27 individual reference genome assemblies will be developed, and then we'll develop again a pangenome assembly based on that, which will be very much useful and reliable for our all genomic studies. Next slide, please. And what we are doing from those 15,000, we'll make diversity panel, let's say about 1,500 lines from 15,000. I mean to say just 10%. And then what we'll do, we'll phenotype for different traits that we need in our breeding programs in Asia and Africa, and only around 200 will be selected for using as a parent or trait donors in different breeding programs. So in order to like have a diverse panel of 1500 for each breeding program based on the traits. Suppose if you need 10 traits in your breeding program, based on the traits, you can have trait frequency higher so that the panel is equally good for the genome wide association analysis. Because what we are facing difficulty, the current diversity, smaller diversity panels, they are biased. There are really few genotypes with the trait that we want. That means those traits, they are going in minor allele frequency. So those are not good actually for the association analysis. So we are trying actually here, like said, developing iterative diversity core panels so that that can be used in our breeding programs for the future. Next slide, please. And this is again, the concept is same what I told as it is done in humans. We were trying to replicate in pigeon pea first, and then maybe in groundnut in the next. Next slide, please. Now, what I'm saying here, understanding in-depth genetic diversity is important from the gene bank. So we can manage the gene bank conservation as well as the diversity. Once, even like it is, if you see genomic resources, so we have like these genome assemblies, but pangenome graph, and all those reference genome, those are required. And then defining core diversities, and I was talking about for different breeding programs, that is very much important because we need the best of the best genotypes as a donor, and to breed the future ready crops. I mean to say climate change keeping in mind. And the most problem that will be coming up is data integration, because the when we are generating huge data, it's always difficulty to analyze them, store them also. Next slide, please. We are organizing one conference that is for groundnut. If some of you are interested, we are organizing in March 2025 in Goa. So you can please go ahead for registration, and you will be having like say international, I mean to say participants there to interact with, and also like this will be one of the good gathering if any one of you interested. Next slide, please. Thank you.
चैट बॉक्स ऑन कर दीजिए, यदि किसी पार्टिसिपेंट्स की कोई क्वेरी है इस टॉपिक को लेके तो वो चैट बॉक्स में लिखेंगे, हमारी मॉडरेटर उस क्वेश्चन को पढ़ के सर तक पहुंचाएंगी और सर उसका रिप्लाई करेंगे। तो जो पार्टिसिपेंट्स किसी तरीके की कोई डाउट है तो आप पूछ सकते हैं।
और अंकुर, जो यहाँ पे नहीं भी पूछ पाते हैं, आप टेक्स्ट बॉक्स में मेल आईडी
मैं
दे
ईमेल
सकते
लिख
हैं।
दूंगा सर। जी
हाँ,
email ID.
suppose if somebody is interested, so I'm very much open to answer. And my apologies for this internet connection. So I don't know why it was happening today, but
अरे नहीं सर, हो जाता है, ये टेक्निकल ग्लिचेस होती रहती है।
yeah.
एडमिन महोदय, चैट बॉक्स ऑन कर दिया आपने?
यस सर।
हाँ ठीक है, पार्टिसिपेंट्स अपनी क्वेरी लिख सकते हैं चैट बॉक्स में। साथ ही साथ मैं ग्रुप में सर की ईमेल आईडी भी दे दूंगा, जो पार्टिसिपेंट्स का ग्रुप है, तो यदि किसी को लगता है तो सर से वहां बात कर सकते हैं ईमेल पे।
और स्लाइड अब शेयर करना बंद कर सकते हैं इफ यू
जी हाँ, स्लाइड शेयर मोड हटा दीजिए आप। जी लेक्चर में कोई गुंजाइश ही नहीं रही किसी तरीके की, लेकिन कोई क्वेश्चन आया है। नंदिनी, आप सुन पा रहे क्वेश्चन को? पढ़िए।
हाँ जी सर, that actually is not a question but appreciation. People are appreciating the valuable knowledge, sir.
एक क्वेश्चन आया है
We
डॉक्टर
have got
कुसुम
a
पटेल जी
question.
का।
Why genetically modified crops are not allowed for consumption in India? From Kusum Patel.
Okay, so you know that this GM crops issue is basically more societal, and now not just scientific, more of societal and then political issue. And this is like a decision we have to take which crop we have to consume, and then how stringent criteria we have to make. So I think I don't have particular answer for you. So this answer is mostly I will leave for the government and then government, I will say.
Okay, next
Yeah.
please.
There is some question related to peanut allergy, I guess.
Yes, sir. Some people are allergic of peanuts. What are those allergy compounds, and can it be removed by using markers?
Yeah, I think this is good question. Just I will take 30 seconds on it. So we actually at ICRISAT, we were not working on peanut allergens, and what we have seen actually that in recent years the peanut allergy you see only in US, Australia, Europe, and all those countries. But you won't see much report is not there from India and China and all those places. Maybe there are cases, but we don't have actually data that how much people are allergenic to peanut. But what I have seen especially that the kids which are grown in US or Europe, and when they are returning, even our NRIs, basically the Indians, back to India, because they have the peanut is not been like say introduced in the childhood, so they are becoming allergenic also. So that's like one of the reason I got it. And in India, mostly why we are not much peanut allergenic, basically we from the right from childhood we consume a lot. But if you come to asking question about markers, we have done analysis. Actually, if you see, we have germplasm. In the germplasm, the lines which are having very less peanut allergens, like proteins. So if we see that in India it is a problem, definitely it can be take up. But as of now, I don't see any priority at the national level in India. But for sure, this is a major issue in European countries.
Next, please. रोहित जी आप पढ़ दीजिए उसको.
Yes, sir. So thank you so much, sir, for answering the question. Though we have one more question, and the question is how the potential risk of GM crops to human health is determined, sir.
See, actually, I think we are asking too many questions. We are putting too much questions on transgenic crops. It's very difficult actually to assess for any crop, even if you develop a new variety or anything. It's very difficult to assess each aspect of human health. And at in our government level, there is set criteria for assessing transgenic crops for human health. I think that is good enough. So I think if it is passed, it should be released. And I will just want to also share, I was reading one of the newspaper that even if the transgenic crops are not allowed to grow in India, when we are importing products from outside, who knows that the raw material used in, suppose let's say in US, there it is allowed GM crops, so the raw material is used for making those products and we are consuming. And there has been in a report somewhere I seen in newspaper that already some of the such products are being detected where the traces of transgenic crops are being found, although it is not allowed in India. Okay, in nutshell, what I'm saying, we need to have stringent criteria, but also little bit we have to take tough decisions for the food security in future. After assessing it, I don't see so much risk or any risk as of now. I have never seen a report of any human health from transgenic crops per se. So I think we have to just cross check and see how it works. Anyhow, if India does not allow also, this is growing in different parts of the country. It is a matter of choice for us as a society, as a government. We can take a good decision on this, maybe with cautious, but we have to move a little bit forward in that one. I'm not sure if I answered the question.
Well, thank you so much, sir. And I think you have solved all the queries. Though we are getting greetings, nice presentation, good presentation, sir. So, Ankur, sir.
हाँ जी. वैसे लेक्चर बड़ा सेल्फ एक्सप्लेनेटरी था सर का. हमने पीपीटी पूरा देखा, सर ने पीपीटी भी शेयर किया है. कुछ पार्टिसिपेंट्स का आग्रह है कि क्या हमें पीपीटी मिल सकती है, तो यदि आप अलाउ करेंगे तो हम रजिस्टर्ड पार्टिसिपेंट्स के ग्रुप में पीपीटी भेज देंगे सर।
Sure, please.
जी जी जी।
And mail ID
बहुत-
also, if somebody, I think in first slide it is mail
Nandini.
ID is there. And no problem. And if I will be happy to interact or discuss with anyone after this meet, not a problem.
जी जी जी, बहुत बहुत धन्यवाद सर। मैं यही कहूंगा कि मेरे आग्रह पे, मेरे निवेदन पे आपने ज्वाइन किया और अपने बेशकीमती समय से जो है हमारे पार्टिसिपेंट्स को अपना समय दिया। बहुत बहुत धन्यवाद सर। आगे भी इस तरीके के एकेडमिक कार्यक्रम में हम आपका इन्वॉल्वमेंट, आपका पार्टिसिपेशन जरूर चाहेंगे सर। एग्रीमेंट
Sure,
फाउंडेशन हमेशा इसी तरीके की एकेडमिक एक्टिविटीज करता है, तो आपका सहयोग, आपकी ब्लेसिंग्स, आपको पार्टिसिपेशन जरूर चाहिए होगा। बहुत बहुत धन्यवाद सर।
Thank you. हमारा सपोर्ट ऑलवेज आपके साथ में है।
जी, थैंक यू सो मच। ठीक है, सभी पार्टिसिपेंट्स लेफ्ट कर सकते हैं। गुड नाइट।
Thank you.